Hey everyone, welcome to my channel. Today we are going to study about apoptosis. Before proceeding ahead, we should understand what actually is apoptosis and why does it occur. We all know that a normal cell exists in a state which is called homeostasis, where there is maintenance of constant internal environment of a cell, and this is the healthy state of a cell. But if the limits of the adaptive responses are exceeded, or if the cells are exposed to injurious stimuli, there occurs a sequence of events which is called cell injury. Now, this injury can be reversible injury, or this can also further proceed to irreversible injury, depending upon the severity of the stimulus and how persistent it is. So, if the injurious stimulus is severe and it is progressive, ultimately reversible injury becomes irreversible injury, and that ultimately leads to cell death. Now, this cell death can be in two forms. Either it can be necrosis or it can be apoptosis. We are going to talk about apoptosis today. So, apoptosis is a pathway of cell death. That's what we saw in the previous flowchart. Here, the cells activate enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. That is what is a cell death. So I told you there are two forms of cell death. One is apoptosis and one is necrosis. We are going to talk about apoptosis. So let's go to the word origin of the word apoptosis. So it's a Greek word which means the process of leaves falling from trees or petals falling from flowers. So the word apoptosis, if you break down, it becomes apo plus tosis. Apo means away, off or apart, and tosis means falling. So that's how the word apoptosis originates. Now, one thing that usually students have in mind is that apoptosis is always pathological, but this is not true. You should think over it and you will try to understand that yes, apoptosis is not always seen in pathological conditions only, it is physiological as well. So ask yourselves this question: is apoptosis seen in pathological conditions only? No, it can be seen physiologically. These are the examples. During normal development of an organism, we all know that some cells die and then they get replaced by the new ones. In mature organs also, the highly proliferative and hormone responsive tissues undergo cycles of proliferation and cell loss. That means there is occurring cell death as well simultaneously, that are often determined by the levels of the growth factors. And in these situations, what did you observe? That there is cell death, and this is always by apoptosis. That means apoptosis is not always pathological. Now, why is this required? I said apoptosis can be physiological as well. So whatever occurs physiologically, that has some meaning, that has some purpose. So what is the purpose of apoptosis that occurs physiologically? It ensures that the unwanted cells are eliminated without eliciting potentially harmful inflammation. We all know that if cells are not eliminated and they persist, then obviously they will induce inflammation, and if inflammation occurs, a lot of damage can also occur if it is chronic. So in the immune system, apoptosis eliminates excess WBCs which are left at the end of immune response, as well as the lymphocytes that recognize the self-antigens and could cause autoimmune diseases if they were not removed. See, in the immune system also, whenever there is end of the immune response, the WBCs don't stay there for long. We all know that once the immune response is over, the WBCs subsequently they undergo apoptosis. Now that is because their work is over. We don't want excess WBCs to stay there and to cause further tissue damage or whatever after our function is performed. That is, immune response has been performed. And another example is that we know that there are some lymphocytes in the body that can recognize the self-antigens, and if these are not destroyed, they will pose the harm for autoimmune diseases in the future, and we don't want that to happen. So body has a protective mechanism against it, that is, it recognizes these lymphocytes and causes apoptosis. So basically what we discussed till now is that apoptosis can also occur physiologically and apoptosis can also occur pathologically, that we'll discuss in subsequent slides. So till now we have discussed that it occurs physiologically. So talking about few examples of how it occurs physiologically, let me give you one example. that is cell turnover. Cell turnover in proliferating cell populations. For example, if you talk about the immature lymphocytes in the bone marrow and thymus. So cell turnover is going on in cell populations which is proliferating at a huge rate. So one example is the immature lymphocytes in the bone marrow and the thymus. And this turnover means that cells are proliferating and they are dying. So that is called cell turnover. Then another example is the removal of supernumerary cells. Supernumerary cells, that is, if they are excess than what is required during development. This is one example. That is, if there are excess cells during development, they are removed. You all know there is a term known as organ sculpting, right? And we all know that now we have, as we develop in the fetal life and the whole process of the embryogenesis and the organogenesis and all this development, it takes place. Then in that there are a lot of changes that occur. So in that, apoptosis is a very important phenomenon, because that is what is responsible for removing the cells that are not required when we are born. Like earlier, we have the tissue present between the fingers that gives the webbed appearance, but then it gets removed by what is known as apoptosis, because now we see that when we are born, the fingers, they are completely separated. So this occurs by apoptosis. Now, apoptosis in pathological conditions. Apoptosis eliminates the cells that are damaged beyond repair. This is seen where there is severe DNA damage. For example, after exposure to radiations and cytotoxic drugs. It is very important to eliminate the cells that are damaged beyond repair. I'll tell you why this is important, because if there is severe DNA damage and if the cell keeps on proliferating, then that means that this damaged DNA is going to the daughter cells, and these daughter cells, when they are dividing, they are giving in turn to their daughter cells, and this cycle is going on. That means we are creating a population that contains damaged DNA, and damaged DNA means there are high risks of developing problems like cancer, because the damage in the DNA can be at a site, at a particular point in the DNA, that can lead to their uncontrolled proliferation or some other problems. We don't want that to pass to the progeny, and that is why if the cells feel that they cannot, so whenever the DNA is damaged, there occurs a process that is called repair. That's body's protective mechanism. But if the repair also fails to these repair mechanisms fails to repair the damage, then apoptosis is one thing that is there in the minds of the cells, that we have to do it because DNA is severely damaged and we cannot take risks with the daughter cells. So at that place, the cells get died, and once the cells die, they can't pass it to the progeny. Hence, apoptosis is a protective mechanism here. Then second is accumulation of misfolded proteins. It also triggers apoptotic death. We all know that the accumulation of misfolded proteins is harmful for the cell. It's important for the cell to die, otherwise it will be dangerous. Certain infectious agents, particularly some viruses, induce apoptotic death of the infected cells. Now that means that body is trying to protect other cells from getting the viral infection. So whatever the cells get infected, they are going to undergo apoptosis. Another example I would like to add here is that if there is duct obstruction, then there can be pathological atrophy in parenchymal organs. This is seen in, for example, parotid gland. So if the duct gets obstructed, then there can be pathological atrophy of the parenchymal organs like parotid gland, and this occurs by apoptosis. So it can be observed here as well, apart from the examples which I discussed earlier. So little bit about what actually happens to the apoptotic cell. The plasma membrane remains intact. The plasma membrane of the apoptotic cell remains intact, but the membrane is altered in such a way that the fragments, which are called the apoptotic bodies, they become highly edible. And because of this they get phagocytosed, leading to the rapid consumption by the phagocytes. The dead cell and fragments are cleared with little leakage of cellular contents, and that is why there is no inflammatory reaction. So apoptotic cell death does not elicit an inflammatory reaction. We'll talk about the morphology of the apoptotic cell in detail in another video. This is a short description of what actually happens. You can see this is a normal cell, this is the normal nucleus, this you can see normal mitochondria, the normal plasma membrane and everything. But what happens here, you can see the plasma membrane is intact, but what you can see is there is condensation of chromatin, the membrane blebs are formed, and then you can see apoptotic bodies which are formed. There is cellular fragmentation, and you can see these apoptotic bodies that are formed. This is apoptosis, and ultimately the phagocytes come and eat these apoptotic cell fragments as well as the cells themselves. Now coming on to the actual biochemical pathways by which the apoptosis actually occurs. So apoptosis is regulated by biochemical pathways that control the balance of death and survival-inducing signals, and ultimately the activation of enzymes which are called caspases. Now what are caspases? Caspases, as the name suggests, there is ases. That means it's going to have some kind of lytic function. So they are cysteine proteases. So how do you remember it? The letter C, right? It's common. Caspase, cysteine. And ases, as I told you, lytic, so proteases. They are cysteine proteases. They cleave the proteins after the aspartic acid residues. That is why you see ASP here, that is for aspartic acid. So they cleave proteins after aspartic acid residues. Now two distinct pathways converge on caspase activation. One is the mitochondrial pathway and the other one is the death receptor pathway. Now one thing to keep in mind is that although these pathways can intersect, they are generally induced under different conditions and involve different molecules and serve distinct roles in physiology and disease or pathology. So we'll be talking about two pathways one by one. First will be the mitochondrial pathway and second will be the death receptor pathway. So the mitochondrial pathway. We all know that mitochondria are remarkable organs. They contain proteins like cytochrome C that is essential for life, but at the same time cytochrome C acts as a double-edged sword. So we know why it's essential for life, because it participates in a very important pathway, that is the respiration. Basically this is the one that is helpful for our body, but some of the same proteins, when released into the cytoplasm, that means the cell is not healthy. In those cases, it initiates apoptosis. So rather than participating in oxidative phosphorylation as it enters the cytoplasm, it causes apoptosis. So when it is there in the mitochondria, it will perform a good function, that is it will participate in the oxidative phosphorylation and produce ATP, and ATP is energy for us, and so it is good for the cell. But once it enters the cytoplasm, there it initiates apoptosis. The release of these mitochondrial proteins is controlled by a finely orchestrated balance between pro and anti-apoptotic members of the BCL family of proteins. So let's understand what is the BCL family of proteins. So BCL2 family of proteins is divided into 3 groups. Anti-apoptotic is the first group, which includes BCL2, BCL-XL and MCL1. They possess the 4 BH domains, so they are called BH1 to 4. They keep the mitochondrial outer membrane impermeable. That is why there is no leakage of cytochrome C into the cytosol as long as these anti-apoptotic proteins they are present, and the pro-apoptotic proteins they are in a state of inhibition, or in a way they are not produced. But this is the case where anti-apoptotic factors are preventing apoptosis. But what if the BCL2 family of proteins promote apoptosis? Then that is the group which is called pro-apoptotic, the backs and the back. So how do you remember it? Backs with the word axe and back with the bark of the tree. So remember, pro-apoptotic means it's promoting apoptosis, means promoting death. So that you can remember with an axe and the bark. So axe cuts the tree, and that's a kind of death. So that's how you can remember. This is just to make you remember. It contains first 3 BH domains, BH1 to 3. On activation, they enhance the outer mitochondrial membrane permeability. Regulated apoptosis initiators are the third group, which includes BAD, BIM, BID, PUMA and NOXA. They contain only 1 BH domain, and that is the third one. Out of all the 4 BH domains, it's the third one that is found here. So they're also known as BH3 only proteins. So yes, this is BH1 to 3. The previous one was BH1 to 4, and this is BH3 only proteins. They initiate apoptosis when they are upregulated and activated. So that's what I was saying, that if they get these regulated apoptosis initiators, when they get upregulated, they are going to initiate apoptosis. So there comes the role of pro-apoptotic factors, and that's why apoptosis is seen. Now this is a very good picture from the Robbins, and this explains everything very well. So 2 cases are shown, A and B. A is a viable cell where there is survival signal that can be in the form of growth factor. Now till survival signal are present, there is no apoptosis. Now how is that possible? So this is the mechanism. When there is the presence of survival signal, there is production of anti-apoptotic proteins like BCL2, BCL-XL and so on. Now this is the mitochondria, and these are the cytochrome C. So you can see the cytochrome C is not leaking into the cytosol, it's still present in the mitochondria. And this blue colored channel, as you can see, is the BCL2 or BCL-X. But what happens when there is lack of survival signals, or when there is some form of injury like irradiation? So this causes DNA damage, and lack of survival signals both activate the sensors, which are BH3 only proteins, and they cause the antagonism of BCL2. So this is shown very clearly in the form of a cartoon, as you can see. Suppose these are the BH3 only proteins, and as I told you, these blue colored channels, these are BCL2. So it goes and you can see attaches to the BCL2. So this is shown in the form of cartoon. So basically it causes, what you have to remember is that it causes antagonism of BCL2, and what you can see is there is activation of the backs and back channel. You all know that backs and back, these are the pro-apoptotic proteins. So when there is activation of these channels, cytochrome C can easily pass out of the mitochondria and enter into the cytosol. So when there is leakage of cytochrome C, there is activation of caspases, and which ultimately leads to apoptosis. This ultimately leads to apoptosis. So you can see synthesis of BCL2 and BCL-X is also dependent on the survival signals. Here there is lack of survival signals, so BCL2, though there is antagonism also, but at the same time BCL2 and BCL-X, their synthesis is also decreased because there is lack of survival signals. Okay, now coming on to the step of when the cytochrome C, after coming out of the mitochondria, enters into the cytosol, what actually happens. So once released into the cytosol, cytochrome C binds to a protein which is called APAF1, apoptosis activating factor 1. After combining with this, it forms a wheel-like hexamer which is called the apoptosome. This apoptosome is the one that binds to caspase 9. Caspase 9 is the critical initiator caspase of the mitochondrial pathway and the enzyme which cleaves the adjacent caspase 9 molecules, thus setting up an autoamplification process. So one thing you have to remember here is that caspase 9 is the initiator caspase of the mitochondrial pathway. Other mitochondrial proteins are also there, like SMAC and DIABLO. Enter the cytoplasm, they bind to and neutralize the cytoplasmic proteins which function as physiological inhibitors of apoptosis called IAPs. So IAPs are basically proteins that they inhibit the apoptosis. They are physiologically present in the body and they are physiological inhibitors of the apoptosis. Now if we want apoptosis to occur, then obviously we don't want IAPs to function. So there comes the role of SMAC and Diablo that neutralize IAPs. The normal function of the IAPs is to block the activation of caspase, including the executioners like caspase 3, and keep the cells alive. So thus the neutralization of these IAPs permit the initiation of a caspase cascade. Next comes the extrinsic or the death receptor initiated pathway. So the previous pathway that we discussed was the mitochondrial pathway. It is also called as the intrinsic pathway. And the second pathway, that is the extrinsic death receptor initiated pathway of apoptosis. In this is initiated by engagement of plasma membrane death receptors. So on the plasma membrane there are present death receptors which are members of the TNF receptor family, the tumor necrosis factor receptor family, that contains a cytoplasmic domain which is involved in protein-protein interactions, that is called the death domain. So basically there is a death domain which is the cytoplasmic domain which is present in the cytoplasm, and this is essential for delivering the apoptotic signal. So we'll see how it is possible. The best known death receptors are the type 1 TNF receptors and a related protein called Fas or CD95. The mechanism of apoptosis induced by these death receptors is well illustrated by Fas, which is a death receptor expressed on many cell types. So as I told you, there is something called as Fas, which is a death receptor expressed on many cells, and it has got a ligand which is called Fas ligand. So when Fas ligand comes, it binds to the Fas, and 3 or more molecules of Fas then are brought together, and their cytoplasmic death domains then form a binding site for an adapter protein that also contains a death domain and is called FAD, that is Fas-associated death domain. So this adapter protein is FAD, which is called Fas-associated death domain. So when Fas ligand binds to Fas are brought together and the cytoplasmic death domain form a binding site for this FAD, and this gets attached to the death domain, and it itself contains a death domain. So FAD that is attached to the death receptors in turn binds an inactive form of caspase 8, and caspase 8 is the initiator caspase of the extrinsic pathway or the death receptor pathway, again via death domain. So multiple pro-caspase 8 molecules are thus brought into proximity and they cleave one another to generate active caspase 8, again like autoamplification. The enzyme then triggers a cascade of caspases by cleaving and thereby activating other pro-caspases, and the active enzymes mediate the execution phase of apoptosis. One thing to note is that this pathway of apoptosis can be inhibited by a protein called FLIP. flip binds to procaspase 8, but it can't cleave and activate the caspase because it lacks a protease domain. So just goes, this flip goes and binds to procaspase 8, so that procaspase 8 is not converted to caspase 8. That's it. Because it binds to that site, so nobody is going to activate it because the site is occupied. Some viruses and normal cells produce flip, and they use this inhibitor to protect themselves from Fas-mediated apoptosis. So this again is a picture of Robbins. This is the Fas ligand and this is Fas. This is the Fas ligand. This is Fas. And as you can see, there is one Fas, but when Fas ligand binds to it, many Fas molecules come together, and this red color shown here is the death domain. A lot of death domains come, and FAD, this is FAD, which is attached to the death domain, and it in turn activates procaspase 8 into active caspase 8, and which further activates the executioner caspases and which promotes apoptosis. So what is the execution phase? Executioner caspases are caspase 3 and caspase 6. So caspase 3 as we did in the intrinsic pathway. Once activated, these cleave an inhibitor of DNase. So if the inhibitor of DNase is cleaved, then there will be DNA degradation. Also, it causes proteolysis of structural components of nuclear matrix, leading to fragmentation of nuclei. So thank you for watching the video, and also try to read it from the book of Robbins, which is really a good book, and this topic is explained very well in that. I hope this video helps you in understanding the topic very well.